This video is a Keystone review for Anchor 2, the chemical basis of life, which includes a pretty wide variety of topics. One of those is describing the unique properties of water and how water supports life on Earth, to describe and interpret relationships between the structures and function at various levels of biochemical organization, so that's going to include atoms and molecules, and explaining how enzymes regulate biochemical reactions within a cell. To start, let's look at atoms themselves. Atoms consist of protons, neutrons, and electrons. The protons and neutrons are in a nucleus. The electrons surround that nucleus. Protons are positively charged. Neutrons have no charge, and electrons have a negative charge. You'll notice that the number of protons equals the number of electrons. The positives equal the negatives. You also notice that there are 4 neutrons in that nucleus instead of just 3. Atoms form bonds in order to be stable. That means that their outer electron energy level is full. A covalent bond is where the atoms are sharing the electrons. With an ionic bond, the atoms are either giving electrons or taking them. For those bonds, that outer energy level needs to have 2 electrons if it's in the first level, or any level after that. The octet rule would mean that there's 8 electrons. It's typically how we see atoms and elements on the periodic table. And so the questions that we need to ask and answer are: what's the number of protons, neutrons, and electrons for each of these? What's the atomic mass and atomic number for each one, and what changes to make these isotopes or ions? So let's just look at carbon as an example, and then you can think through the other 2 examples on your own. Starting with the number of protons, the 6 represents the number of protons, which is the atomic number. Because protons are positive and electrons are negative, the number of protons equals the number of electrons. To find the number of neutrons, we take the 6 protons plus the number of neutrons to get the atomic mass, which in this case is 12. So 6 plus what equals 12, and that tells us that carbon has 6 neutrons. The top number is the atomic number, the bottom number is the atomic mass. To make these ions, we need a different number of electrons. So there's 6 protons, but maybe there's 5 electrons or 7 electrons. Either way, that would make the atom an ion. An isotope will have a different number of neutrons. This graphic shows the different properties of water. That water is a polar molecule. That means it has a positive side and a negative side. As a result, water is a solvent, sometimes called the universal solvent. It's able to dissolve things. Water molecules form hydrogen bonds because of that polarity. The positive side of one water molecule orients to the negative side of another water molecule, causing some interesting properties of water. For example, cohesion, where water molecules stick together and form things like drops. Adhesion, where water molecules actually stick to the surface of things. Those 2 properties together cause capillary action, where water molecules can move through very tight spaces. High heat capacity, where that hydrogen bonding prevents the water molecules from moving rapidly as they heat up. And so water takes a long time to heat up and a long time to cool down. And because of that, positives and negatives, the water molecule expands when it freezes, meaning that ice is less dense than water, creating this kind of surface coating of ice where fish could live underneath. These properties, like cohesion, also cause things like surface tension, which allow things to rest on the surface of the water. As mentioned before, water is a solvent. That means it dissolves things. When water, the solvent, dissolves some solute, that makes a solution. Suspensions are when the solute does not dissolve. In fact, it will settle out. This is a copy of the pH scale. Some things to keep in mind when you think of pH. Because remember, water is a solvent, and as a result, it's going to dissolve acids, causing them to release hydrogen ions. The pH scale measures the acidity of a substance on a scale of 0 to 14. 7 is neutral, that's pure water. 0 to 6 are acidic things because the hydrogen ion concentration increases as you go down the scale. Going up the scale, the OH ions increase. 7 is 2 H's and 1 O, H2O, water, which is completely neutral. The scale is a logarithmic scale, so going from one pH value to the next, you have to go by a power of 10. In this case, to go from soap, which is 10, to an 11 is 10 times more basic. 12 is 100 times more basic. 13 is 1,000 times more basic. Here we have pictures of carbons. Carbon atoms are unique because they can create these 4 chemical bonds with their 4 electrons in that second energy level. These chemical bonds can take the shape of rings, chains, and branches. The carbon ring is illustrated by letter C, the chain is illustrated by letter A, and the branch is illustrated by letter B. The lines between those carbon atoms represent chemical bonds. The double lines means they're sharing 2 electrons instead of just 1. This capacity of carbon allows it to make really interesting complex molecules. We sometimes call them organic molecules. Those molecules, when they're large complex molecules, are called polymers. Polymers are made up of smaller subunits called monomers. So monomer just means one, poly means many. Carbohydrates, lipids, nucleic acids, and proteins are all polymers made up of smaller parts. But let's look at some of those parts, including the elements that make it up. So for example, carbohydrates are made up of CHO, lipids are made up of CHO, nucleic acids are made up of CHONP, and proteins are made up of CHONS. You also notice that lipids have a different ratio between the hydrogens and oxygens than carbohydrates. Lipids have much fewer oxygens than carbohydrates do. But lipids have a carboxyl group, and so do amino acids that make up proteins. Nucleic acids have a sugar backbone, usually ribose or deoxyribose. That sugar is a carbohydrate. Living things use carbohydrates for short and long-term energy. Lipids are more for energy storage. We also use lipids as hormones and sometimes insulation. Nucleic acids store genetic information, and proteins can be useful for growth, maintenance, and repair. Some examples of carbohydrates include monosaccharides like glucose, or disaccharides like lactose, or polysaccharides like starch, cellulose, glycogen. Examples of lipids would be things like triglycerides, which can be saturated or unsaturated. You know they're unsaturated if the molecule bends and if it has a double bond. Lipids could also be used for steroids and cholesterols. Nucleic acids are things like DNA, RNA, and ATP. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Proteins can be things like albumin, hemoglobin, actin. You'll notice that they have an IN ending, which is a giveaway for identifying proteins. Consider these 3 different pictures. We have an amino acid, which is a monomer for a protein. We have maltose, which is a disaccharide, a double sugar. And we have a triglyceride, a glycerol, and 3 fatty acids. Notice some of the similarities and differences between them. For example, the amino acid has a double bond. So does a unsaturated fatty acid. You'll notice that the amino acid has a carboxyl group. So does the fatty acid chain. You'll notice that when these monomers join together to form polymers, they go through a process of dehydration synthesis. That is, at the place of bonding, water is lost to create the bond. Hydrolysis is the opposite, adding water and splitting the molecule. Here's a diagram showing the effect of insulin and glucagon, which are hormones, on blood sugar levels. So you'll notice that as insulin levels rise, that's going to stimulate cells to take glucose out of the bloodstream, lowering overall blood sugar levels. You'll notice that glucagon has the opposite effect. Glucagon targets the liver to release glucose, which causes blood sugar levels to rise. So a diabetic would need to take an insulin shot. Why? To help sugar go from the blood into the body cells that need it. Here's a picture of a cell membrane. Notice the different organic molecules that are present in the cell membrane. We have phospholipids, so that's a phosphate group with 2 lipid tails. we have protein channels, and we have carbohydrate chains. All these things, all these different organic molecules making up the plasma membrane of the cell. This is a picture of a triglyceride. Notice that the unsaturated fatty acid chain has a double bond and is bent. Unsaturated fatty acids are typically plant products in liquid at room temperature. Saturated fats are typically animal products in solid at room temperature. Enzymes are a subcategory of proteins. Enzymes are proteins. They're a catalyst, which means they speed up chemical reactions by lowering the activation energy. Activation energy is the energy required to get a chemical reaction started. Most chemical reactions will happen in your body, but the problem is they don't happen at a speed which is conducive to life. So by enzymes lowering that activation energy, they can catalyze it or speed up the reaction in such a way that it can happen at a speed that helps keep you alive. The normal energy required is represented by the black line. The lowered activation energy level is illustrated by the red line. Here's an example of how enzymes work. You'll notice that there's a substrate. That's going to be some molecule that either needs to be put together or taken apart. That substrate fits very specifically into an active site of an enzyme, forming the enzyme substrate complex. The product then is that substrate or that chemical reaction either breaking something apart or putting something together. If it's putting something together, we call that synthesis, where the two different molecules are brought together at the active site and a new product molecule is built. Digestive reactions are going to take those larger polymers and break them apart into smaller monomers. And the enzyme in that case is going to be digesting those molecules. Enzymes work only in specific ways and only in specific conditions. For example, there's a specific pH range where most enzymes will work. Too high or too low, the rate of the reaction goes down dramatically. And that's because the enzyme is a protein. The outside of that pH range will denature or change the shape or nature of that enzyme. Similarly with temperature, if it's too hot or too cold, the enzyme will denature and it won't work very well in the conditions. Notice that as the substrate concentration increases, the rate of the reaction increases until all the enzymes are occupied. Once all the enzymes are occupied, the only way to continue to increase the rate of the reaction is to add more enzyme. I hope that gives you a brief overview of Anchor 2, the chemical basis for life.